So the function of zonal adherens is again nothing more than just connection of the cell, connecting the two different elements of the cells, of the two different epithelial cells. This zonula, this zone, is controlled by a very large group of proteins called cadherens. And actually the name is very easy to remember. Adherens, zonal adherens, the adhering junction contains cadherens, adherens again, but with a C in front. The point is that why do we care so much about these? Because they play a role into the malignancy and cancer invasion. How does it actually work? How do we understand this thing? Well, because if you lose, generally speaking, the malignancy is one of the components, the typical components of the cell functions, of the cancer, of the malignant tumors and malignant cells, is the invasion from the top. For example, let's take an epithelial cancer. Okay, let's take that when the top of the cell, the epithelial cell, becomes cancerous, and the tendency of the malignant cells have the tendency to invade into deeper portions of the structures of the cells. For example, they tend to invade towards the subepithelial leucinoid tissue. This is one typical feature. So if you lose the functionality, if you lose the structure of cadherens in between these two cells, it allows the cell an easier transition, because there is one less, let's say, inhibiting structure. There is one more structure that inhibits the movement from the basal part, from the proper structure, which is inside the epithelium, and if you lose, the more components you lose, the more, let's say, connections you lose to the other epithelial tissue, the easier it is for you for the cell to progress and to invade into deeper layers. So that's why there's this comment that it participates in malignancy and cancer invasion, because of exactly this function, because it adheres and maintains the structure location, the location of the epithelium the location of the epithelium exactly in the epithelium, and not invading other structures where it shouldn't be. So these two next structures we'll discuss right now, the macula adherens, in other words called desmosome, much more common name, desmosome, and the nexus or the gap junctions are more sparsely located. Again, typically always we're going to find at the top the tight junction, exactly beneath it the adhering junction, and now the desmosomes and the gap junctions can be found spread out throughout the whole rest of the lateral sides. So let's see them a bit. What is the function of a desmosome? So the word desmosome comes from the two words desmo and zome. Desmo means connection, and so means body, means practically a component, a structural component. So the desmosome is the binding and the connecting structure. It actually binds intermediate filaments of both cells, of two cells. If we have two cells on the side like this, the one epithelium which contains intermediate filaments, which serve the function of cell maintaining and strengthening the cell structure. So these intermediate filaments will be bound to specific points on the lateral points, specifically the desmosome. And now this desmosome is going to be the one that connects the other cell, that both intermediate filaments are actually connected on this binding point of the desmosome. So in this case, again, let's take a look at the picture. These orange fibers are the intermediate filaments. So we can see that the desmosome practically combines and connects the two different intermediate filaments of the two different cells. So you can see these ones are exactly the intermediate filaments of the left cell, and this is of the right cell. So this is the point of adhesion. So the desmosome practically connects these two cytoskeletal components, the two cytoskeletal parts of two different cells, hence allowing the very, very tight connection of these cells. It practically combines and attaches the cytoskeletal components of two different cells. We can find this structure also not only in epithelia, but also can find them in the heart or other structures as well. Typically, for example, is the cardiac muscle cell. We can find these desmosomes again in the cardiac muscle cells. Let's talk a bit about the gap junction. The gap junction is a very, very important structure that also is found in more than just epithelia, but this is the first example that we'll see, and the more typical example where we'll see. These are molecules that are hexamers of connexins. So the gap junction is, imagine like a pizza-shaped structure with 6 components with 6 parts. Each pizza part is going to be called a connexin. So there is 6 parts of connexins that form this channel of this gap junction. What is the point? So what? You created practically opening channels from one cell to the other. So what? Well, the point is, and the consequence of this channel is that you can actually exchange cells. You can exchange molecules from one cell to the other. Of course, these molecules are small, typically beneath 1,500 mR, meaning, for example, ions, very small saccharides, very small maybe amino acids, tiny structures that have that can actually be moved from one cell to the other. This is a very important structure, and most importantly, this actually structure is responsible for the heart beating because we need to have a synchronized heart beating, and the passage of ions from one cell to the other in a specific time is what allows the heart to beat in synchronicity. In the cases of epithelia, of course, it plays a different role. It plays again the role of ion movement, but is of not the just major importance. Of course, again, it's also important, but less, of course, when it comes to the cardiac function cells. So this is the point of the gap junction. Lastly, the hemidesmosomes. These structures are found in the basal part of the cell. So we already talked about the tight junctions, the adherent junction, the desmosomes, and the gap junctions. So last one, we find the hemidesmosome. Hemi again means half, and desmosome, we already discussed about the desmosome. So if you actually think about just the word is enough to give you the structure, to give you the function of the structure. We find that this structure, the hemidesmosome, is the binding feature, the connecting feature of the cell to the basal membrane, or to the basal lamina as well, because it actually connects with the basal lamina. So the hemidesmosome is the structure that connects the cytoskeletal components, the intermediate filaments, to the basal lamina, and allowing the cell to be tightly adherent and specifically stable on the basal membrane. So the elements, the many components of the basal lamina connect with this hemidesmosome, hence structuring and hence securing the location of this epithelium on top of the basal membrane. As I said, already mentioned right now, always beneath the epithelium, we're going to find this structure of the subepithelial loose connective tissue. Sub means beneath, and epithelial is, of course, beneath the epithelium, loose connective tissue. We'll find this structure, we'll just talk about this structure way more in the connective tissue video, but at this point, just have this general knowledge that always beneath the epithelium, we'll find this, the lamina propria. And whenever you hear the term lamina propria, it's the same term as the subepithelial loose connective tissue. So let's see actually the first example, the simple squamous epithelium, what it is and where we can find it. As I just said before, typically the squamous epithelium has the function of exchange, and the very nice example to see this function is with the lung, with the pulmo. So let's actually see these structures. Here we find this is the first section of the pulmo. So once we zoom in the center, we'll find actually in the lung we're going to find twice the simple squamous epithelium in two different locations. One is in the pleura on the side, on the very actually border of the structure of the lung. And of course, we're going to find also in the center the alveolar cells, the pneumocytes. These pneumocytes, of course, the function of these pneumocytes is to exchange gas, to exchange blood, to exchange within the blood carbon dioxide and oxygen, of course. So let's actually see them in the microscope first. So the first structure we're going to see first is the pleural cavity, the pleural side. So in this lining here, this is the visceral pleura. There are two different layers. There is the parietal layer of the pleura and the visceral layer of the pleura. The parietal is towards of course, the outer part, and the inner part is the visceral part. So here we can see the visceral part. So here we can find the simple squamous cells, actually these ones, that are responsible for the production of a very viscous fluid, of a liquid that actually is found within the pleural cavity, and the function is, of course, to reduce friction, to practically prevent the lung from being a very sensitive organ, from being somehow damaged by the hard rib. So by practically allowing this liquid to be within the two different surfaces of the pleura, the parietal and the visceral layer, the outer and the inner, and within these two cavities, within this cavity, we can find this liquid. So because this liquid is high in component in lipids and other type of proteins that reduce friction, it allows for the lung to slide in through the rib and not be compressed or somehow mechanically irritated by the rib. So this is the first example. And we can find here, see the morphology of the simple squamous epithelium, a very flat-like and compressed cell in this manner. It looks like this, and this is the first example. Let's see the other example, which is the pneumocyte, the type 1 pneumocyte. So here we go. Here we can find the typical squamous epithelium, the typical appearance of the simple squamous epithelium. Again, this is called the type 1 pneumocyte, where it discusses its function again exchange of gas. So next up, we're going to find the simple cuboidal epithelium. Just in comparison, we saw so far the simple squamous epithelium, which is a flat-like structure, and the nucleus always, generally speaking, this is a general histological rule. The appearance of the nucleus is typically proportional to the cell size and cell shape. Meaning what? Meaning that in the simple squamous epithelium, this is the structure of the cell, the nucleus will typically have again this shape of nucleus. So it's proportional. This is a very, very helpful information, a very helpful tip to help us distinguish which cell is overlapping and which cell is just the true cell that we can find. We'll see this example later on and why actually it's so important. So let's move on to the cuboidal epithelium. One typical example of cuboidal epithelium is in the kidney. Of course, in the kidney, the function of the kidney is filtering of the blood, clearing of the structure, clearing of the plasma, and of course to absorb some ions and to secrete some ions as well. So the typical function of the cuboidal epithelium in the kidney is absorption and secretion. So let's actually see the structure. So this is a typical macro picture of the kidney zoomed out, and if we zoom in, we can find this is a typical structure called the glomerulus. We'll discuss the structure in the histology too. But the point, just at this point, just use the glomerulus as a reference, as a point of reference. Surrounding the glomerulus, we're going to find many different ducts, many different structures, so here we can actually see that these ducts are lined with a cuboidal epithelium. As we can see, the cuboidal epithelium has this cube structure, and of course the nucleus is spherical, again proportional again to the cell shape. Let's actually see this in the microscope, shall we? So this is again, as we said, the zoomed out picture. Let's zoom in and find a glomerulus. So again, this is the typical appearance of a glomerulus. As I just said, surrounding it, we're going to find many different tubules, and of course, most of them are practically, with very few exceptions, most of them have this lining of simple cuboidal epithelium. Let's zoom in a bit more and see the morphology. Again, we can find the cuboidal cell and a very, very spherical, typically spherical nucleus. And this is how the cuboidal epithelium looks like typically. Next up, we're going to find the simple columnar epithelium. Now we first see the term microvilli, the first term. We'll discuss what this is. This is our first example of a special apical specialization of a cell. So let's actually first up see what the columnar cell looks like, and then we'll discuss about the microvilli. The columnar, of course, this is one of the very easy organs to actually distinguish and to see the columnar cells is the gallbladder. So this is, of course, the macroscopic, the zoomed out picture of the zoomed out picture of the gallbladder. And once we zoom in on the mucosal layer, this is the mucosal layer. So we focus on the top. How do we actually wipe the top? Typically, this is a covering epithelium. So typically, the covering epithelium will be found in the top, on the one side or the other side. So because of, again, the function, this is the typical example of a covering epithelium. If it was a glandular epithelium, we could find them within the structure, within the section. So not on the sides. So let's actually focus on the top part to see this epithelium and what it looks like. So we're going to zoom into this bridge, this epithelial cells. As we can see here, we can find the tall cell. It's a very tall cell. This is the basal membrane right here, beneath it, exactly beneath it. The basal membrane is a structure not visible through the light microscopy because it's very, very thin. To see this structure, we have to use the electron microscope. So always there is imaginary line that is the basal membrane. So let's zoom in, let's see these cells. Again, the morphology is a very, very tall cell. It looks like a column, hence the name columnar. And again, the cell's nucleus is proportional to the size, proportional to the shape of the cell, which is like an elongated sphere. So let's see them in the microscope again. So again, this is the zoomed out picture of the gallbladder. We're going to zoom in on the top, actually, on top structures. So as we can see this very, very nice structure, this very clear distinction of this columnar cell. And again, the nucleus is the typical shape, the morphology of the nucleus is sphere, but a bit elongated sphere. And this is so easy to determine and to distinguish the shape of the columnar cell. Now, on top of it, we can find microvilli. Now, let's say these wave-like structures, this wave-like appearance is the structure of the microvilli. what is the function? So, what is the function of macrovilli? What's the purpose of macrovilli? Microvilli, this is the first example of this special apical specialization of the epithelia. So the function of the macrovilli, also called brush border or striated border, is absorption. The point is to absorb structures, to absorb molecules. And this is a very, very nice picture of what it actually looks like. There are wave-like structures, wave-like appearances, and this wave-like structure forms, and not the structure's architecture is there for a reason. Because of, let's just take for example the typical columnar cell, which is again an elongated column-like cell. If you have a straight surface and not any wave-like structures, you're going to have a specific surface area. The moment, let's take for example that it's, let's say, I'm saying a random number, 5 millimeters, okay, which is of course excessively much, but let's say for example 5 millimeters. So if you have a straight distance of 5 millimeters, the surface area is going to be very, very small. If in this case, in the same exact distance of 5 millimeters, you make these wave-like structures, you increase the surface area dramatically. And because the surface area is proportional to the absorption rate, this is why the cells form this structure, to increase the surface area of the top and to allow much more nutrients and much more molecules to pass and to be absorbed in this cell. So this structure is relatively stable. There is no point of movement, which is actually a bit ironical because it's the main structure of the macrovilli is microfilaments, and we discussed already that the microfilaments play typically a role of movement. So in this case, we can actually see this actin filaments, the F-actin, this is the filamentous actin, being along the whole structure of the macrovilli and being anchored to the cell membrane with myosin. Again, this is typically moving structures. And again, the fibrin, which is a cross-linking F-actin. Again, F-actin. So all of the structure that it practically makes up the macrovilli is macrofilaments, which is, again, this is the very typical example of the exception. We see that the macrofilaments typically are mobile structures, but here they play a role into a mostly non-movable structure. This is relatively stable. Minor movements, of course, are given and produced, but that's not the point of macrovilli. And again, the function, of course, of macrovilli is absorption. So, let's move on. Let's go to the epididymis. Now, here we're going to find the pseudostratified columnar epithelium with stereocilia. There are two different new terms we have to discuss: pseudostratified and stereocilia. There are many different layers of cells, generally speaking, in the epithelium. The epithelium can have only one layer. In this case, we can name it simple epithelium. We saw, for example, the simple cuboidal epithelium, simple squamous epithelium. In case of many layers, which means true layers, which means the one layer of cell, second layer of cells, third layer, and so on, this is the stratified. Stratum is again the layer. So stratified means layered epithelium. The pseudo-stratified, the term pseudo generally indicates that something is fake, it's not true. So here we're going to find the first structure, the pseudo-stratified, which means that it looks like there are many layers, but actually there are not many layers of cell. There's only one layer of cells. And we'll see how it actually looks like and how to distinguish this structure. So the second term we don't know so far is stereocilia. Stereo again means solid, means stable. So again, the structure here is cilia. There's again this typical long structure, but here the stereocilia is a different structure. It's a different morphology. It has a different morphology, and the point of the structure again is absorption. But it has an excessive length, approximately 5 millimeters, which is a very, very big number. The number of stereocilia is the length of the stereocilia is actually very, very big. 5 millimeters. So let's zoom in and see the structure. This structure, the first typical example of stereocilia we're going to find is in the epididymis. There are two locations in our body that we find the stereocilia. One is the inner ear, and the second is here, right here, the epididymis, which belongs to the male reproductive system. So if we zoom in, we're going to see this typical structure. This is the columnar cell, the pseudostratified columnar cell. This is the columnar cell as it is in the beginning. We're going to zoom in and see the microscope so far. What is the other cell we find? So it looks like there are many layers of cells. How do we see many layers of cells? Generally speaking, you can find the many layers of cells based on the amount of nuclei. The nucleus is the only true verification that a cell is there, because generally speaking, some points in histological slides are not clear. So your only indication and your only clear indication that there's a cell actually present in this location that you're looking in the microscope is the nucleus. So in this case, we can find the columnar cell, and on top we're going to find this very, very long structure, the stereocilia. Again, the function of stereocilia is again absorption. Let's talk about it a bit more. I talked about the two locations, the epididymis and the inner ear sensory cells. You can find they can be found there. And the structure again is similar to microvilli, which means again they're less mobile. In this case, they would actually call the stereocilia to be immobile structures, completely immobile structures. The difference of the microvilli, of course, is that the length is 5 millimeters, which is very, very big comparing to the size that it is. Let's check out the epididymis in the microscope. So here you can see the testes, and exactly on top of it is the epididymis. Of course, we're going to focus on the epididymis right now. So let's start seeing the cell, shall we? We can see two different nuclei, two different morphologies. We find this cell right here, the small cell. This is called the basal cell, and this is the nucleus of the columnar cell. So because we actually see these two different cells, we can see the basal cell right here and the columnar cell. The first thought is that there are too many layers, there are two different layers. The first layer is that of the basal cells, and the second on top of the columnar, which is false. Why? Because both of these cells are actually one layer. Both of the basal cell and the columnar cell are actually both anchored in the basal membrane, and this is the criterion on which we can actually distinguish and say that this cell is stratified and this cell is pseudostratified. If all the cells are attached to the basal membrane, which means even the columnar cell and the basal cell are attached to the basal membrane, then we can actually find only one true layer. And on this true layer, we can find both the basal cell and the columnar cell. Let's talk a bit about the basal cell. What's the function of the basal cells? Generally speaking, we'll see this structure, this pseudostratified structure, many times in the human body. So the common structure, the common function of the basal cell, which is again this spherical cell, it's a typically cuboidal type, let's say, of cell. The primary function is mitotic generation. These cells are progenitor cells of the columnar cells, which means that if for some reason the columnar cell dies, then the basal cell will differentiate and produce this cell. So if, for example, this columnar cell here in the epididymis dies, then the basal cell will activate itself and actually differentiate into this columnar cell as it is. So generally speaking, all the basal cells have this common function. Next up, we're going to find trachea. And why do we care about so much about trachea? Because we find this unique epithelium, this ciliated, pseudostratified columnar epithelium. Generally speaking, the term, the whole length, that we call ciliated, pseudostratified columnar epithelium is typically also called respiratory epithelium. Why? Because it's typically found in the respiratory system. So the first thing that we're going to see is in the trachea. So let's actually see this structure. If you take up the slide trachea, you would see these structures right here. These are glands. This is our epithelium. We'll discuss about the epithelium just a bit, about the glandular glands a bit later. And now we're going to talk about, this is of course the cartilage. We're going to talk about cartilage in another video, the hyaline cartilage. And here we can actually, on the top, we're going to find the epithelium that we care so much about right now. So if we zoom in, we can actually see clearly this pseudostratified columnar epithelium. How do we know again that it's pseudostratified? Because we can see the basal cells, and because there's only one true layer. Because the columnar cell and the basal cell are both connected in the common basal membrane as it is. Interesting feature of this slide. The trachea, as you can see, has this very, very long structure right here. And in fact, here's where the basal membrane is the most prominent in the whole human body. In fact, this is where it was discovered. The first discovery of the basal membrane was done in the trachea and respiratory system because of its great length, and the biggest size of the biggest basal membrane found in the human body is in the respiratory system. So in this structure we can see the columnar cell, the typical morphology of the columnar cell. So we know that this cell, for example, is typically 100% a columnar cell. So you can see the columnar cell as it is. This is exactly this tip that I gave you before with the nucleus shape proportional to the size shape. This is where it comes handy, very, very useful and very, very handy. Why? Because here you can see a very spherical nucleus. The very spherical nucleus is not the typical morphology of a columnar cell. As we saw before, you can see in the picture also, the typical morphology of such cells is this elongated sphere. And here you can just find a very, very spherical, not elongated, just a spherical nucleus as it is here. So this helps you understand that this part, this nucleus, belongs to a basal cell, that is overlapping in the picture. Never forget, you are visualizing two-dimensional structures, not three-dimensional structures. So it's very, very common to have this overlap of one structure on the other. This does not mean that you can find a basal cell towards the apex. Never, of course not. The basal cell is always located on the basal membrane. But here you can see the very typical example of overlapping, right here. So let's talk about cilia. What is the function of cilia? Cilia are mobile structures, and the point of the reason why we have cilia in our respiratory system is because we need to get rid of always pathogens and foreign substances that are found in the air that we breathe in. So the function of cilia is to move these molecules outside of the respiratory system. And there are two different, let's say, fates of all of these foreign particles. One is that after this movement from the respiratory system, the upwards movement, we can either expectorate them, practically mean that we can cough them out, or we swallow them. These are the two options, the two fates of these foreign particles. This structure, which actually is very, very mobile, is made out of microtubules, which microtubules, as again we said before, typically have a structural function to maintain cell structure and cell shape. In this case, we can see how the microtubules play a role in movement. Here we find a very, very unique structure called the axoneme. So the axoneme is this complex structure that is made out of tubules, microtubules, pairs of microtubules that are connected to central microtubules. These circles right here are two microtubules. Again, these spheres right here, these tubes again are microtubules. So how do we make this structure move? How do we make this array of microtubules actually move? Well, if we zoom in the structure, we can see that these two microtubules are connected with D9 arms. D9 is a molecule, is a protein that actually has one function: to grab and to move and to practically inwards move the other microtubule, the other structure that it actually grabs on. So we have a spherical array of microtubules, and all of them contain D9 arms. So these D9 arms, once they're activated, they will grab on the next pair of microtubule and pull towards on this side. So in the end result of all of these, the structural movements, the structural cells, the structural D9 arms, grabbing and pulling inwards, pulling towards this direction, of course, going to pull towards the center of these, towards this center of the A and B microtubules right here, is going to be just a whole sphere rotating. The question is, even if we have, so okay, we have this movement, we have this rotation, the continuous rotation of the axonema. How does this translate to the up and down movement of the cilia? Because the cilia, all of this structure, this kinocilia, is going to move in this upwards manner. It's going to have this direction. If you imagine that this is let's say the trachea is right here, and the point is to move upwards this structure, this foreign particles. So this movement, how can this up and down movement be produced by rotation? The trick is that these D9 arms are grabbing the others in a specific angulation. So imagine that this pulling motion, this rotatory movement, actually with these D9 arms are in an oblique way, in an oblique direction. So because there is this oblique pulling, you have the up and down motion. You have this. The end result is the up and down motion of the cilia, even though the D9 arms are rotating, rotational forces. But because of these rotational forces, they all end up in a sum of oblique angled pulling and oblique angled rotation. This ends up, and this results in the movement of the whole axonema in a very oblique way, hence leading to this oblique movement of the cilia and allowing for these foreign particles to be expectorated or swallowed. So this is how you get rid of foreign particles from your respiratory system. So the primary cilium. This is a very, very similar structure to the cilia. So the kinocilia. The term kinocilia is better used to actually show that these cilia are mobile. Kino again means movement, production of movement. So we also find the primary cilium. That's why I don't really prefer the term cilia. I prefer the term kinocilia, because their primary cilium is another structure found in different cells, and the point of this primary cilium is just of signal reception, signal production, so and signaling generally speaking. So you can see that these signals can result in detection of light, motion, odor, and so on and so on. So the primary cilium actually is a very, very long structure, even longer than macrovilli, and quite bigger than the macrovilli. So this is the trachea. This is the longitudinal section. This is the cross section. Let's focus in whatever section you prefer, either the long or the cross. It's exactly the same. The epithelium is both found in the top, so our structure of interest will be seen actually no matter what. So we can find the many columnar cells. Of course, they're overlapping. There are not many layers. The true layer again is there only one layer, of course, here. And the basal cells, we have the typical spherical nucleus and are very, very close to the basal membrane. So again, the function of the basal cell is the differentiation to the columnar cells. The cells, of course, the respiratory cells here in this case bear the kinocilia, and these kinocilia produce this rotatory movement to move out, to move upwards the foreign particles that are found in the respiratory system. Let's move on and let's talk about the ureter. What is the ureter? The ureter, what's the special thing about the ureter, and why do we need to discuss it right now? Because the ureter contains this very unique epithelium called transitional epithelium, or also called urothelium. The structure of the urothelium is very, very unique. Why? Because we find specific type of epithelia, unique type of epithelia. And these are the surface cells, the surface epithelium, also called umbrella cells. What's so unique about them? Well, the function of the ureter, of course, is to let urine pass through. There is one issue with the urine, the osmotic pressure and the osmotic concentration of the urine. Of course, because the urine are filled with ions and filled with many, many toxic molecules, typically these molecules are lethal to the cell due to the difference in osmotic pressure. Because we have the high amount of ions in the urine, the difference is that the typical concentration, the ion concentration of the urine and the typical concentration of the cytoplasm, of course, there is a major difference. And as a consequence, naturally speaking, because there's a high difference in the ions, you're going to have movement of water, and the water typically goes from the hypoosmotic to the hyper. And because of this difference between them, these concentrations, because of the osmotic concentration, the result would be that the high amount of the water is going to go from the urine towards the cell, ending up in cell swelling. And as a consequence, because of this extreme difference and because of this continuous in-stream and inward production of the inward aggregation of water inside the cell, you would result into the cell bursting because of the excessive amount of water in the cell. So the unique feature of the urothelium, of this umbrella-shaped cell, is that it has this unique osmotic barrier in the cell membrane that practically prohibits this movement of water freely from the urine to the cell. So this is the unique thing about the epithelium. That's one unique thing about the urothelium. The also difference is, of course, that this structure is pseudostratified. Again, we find all the different cells. The second unique thing about the urothelium is that this structure is truly stratified. We find many layers of cells right here. In fact, we have 3 layers of cells. We have the basal cell again. The function of basal cells is exactly the same as we discussed in the other pseudostratified epithelium. And here, of course, there's the basal cells again. Exactly after them, we have the intermediate cells. And lastly, we find the umbrella-shaped cells right here, or also called surface epithelium. You will find this structure, of course, of ureter being into 2 different manners, into 2 different, let's say, into 2 different forms. When there is urine present, it's going to be compressed, and makes sense because when you have urine present within the ureter, this urine take up a space and, of course, compress the structures that are surrounding this tube, which is the ureter. So when there is urine inside this, we're going to find this morphology of the urothelium, this compressed appearance of the cells, and this is going to be called the fully extended one. And when there's urine absent, when there's no urine in the ureter, of course, It's only natural that these cells because they do not receive any pressure, they're more relaxed and they're more, let's say, open. There's nothing to compress them, there's no force to actually push them down. So this is the fully relaxed form of the urothelium. Let's see them in the macroscope actually. We're going to find them in two different versions, in the cross section and the longitudinal section, as most of the structures. So we're going to find also the cross section and of course the longitudinal section. The cross section, of course, if we cut this stratified layer in the cross section, we're going to find these nuclei being in a centrally placed location and an empty appearance of the cytoplasm. And the long section will actually more easily identify the superficial cells, the intermediate cells, and the basal cells. Here we can see the superficial umbrella-shaped cell, the intermediate cell, and the basal cell right here. Or the basal cell even much better here with a more typical nucleus as it is. Always, when someone asks you to show them a cell, always try to find the more typical picture, not the more in between, not the more, let's say, or the more accurate. Go for accuracy. So typically, we can see that this cell is the most surface one. The morphology of the nucleus is exactly typical of that umbrella shape. It's again elongated, let's say spherical elongated shape. The intermediate cells actually have similar appearance, but they're more deep and typically cross-sectioned. And lastly, we can find the basal epithelium with the spherical structure as it is here in the basal cell. So again, here we can find the long section and the cross section. Let's focus on the cross section. So here we can find the cross section. Again, actually the appearance is the typical appearance of the cross section. We can find the umbrella-shaped cell on the top, the intermediate cells exactly in between, and lastly the basal cell. This is a very nice example of the basal cell right here. Again, the morphology of the nucleus is the one that indicates us clearly which cell is which.